Hello everyone. So this video is about cells and all their parts and pieces and all of the specifics that those parts and pieces, which we refer to as organelles, do and play in the daily living of a cell. So let's jump right in. As usual, feel free to speed the video up, slow the video down, or pause the video as you need to. As always, there will be a break at around the midpoint if you are splitting the video up into two parts or if you just need to have a break for yourself. So here we go. Okay, so these are some quick video podcast style wrap ups that would be a great way for you to quickly review or refresh or just to hear all the information again so that you can have it in a nutshell, if you will. I will try to have those links for you in the lesson that this video is a part of. So we tend not to get into too much history in this class, but there are some biologists who are very important to just even how biology runs today in quote unquote modern times. So when we start thinking about cells and microscopes, two of the names that come to mind are Hooke and Leeuwenhoek. So, and these guys kind of existed in the 1600s. Robert Hooke was the first person to invent a microscope, if you will, and you can see an image of what that microscope looks like on the screen. And funny enough, even when you look at big fancy microscopes, a lot of the layout, the initial parts and pieces are still the same. They just look slightly different. But he was the first person to coin the phrase cell, and he did that by looking at cork, which is a woody material, underneath the microscope. So you can actually see that on the screen as well. And if you look closely, you'll see we have these little compartments that look very similar to what we know animal cells to look like even now. Then there was Antoine van Leeuwenhoek, who invented a microscope as well. He worked upon improving what already existed, let's say, and he observed what we called animalcules, which again were types of cells. We now know they were types of cells, but these two scientists were revolutionary in terms of cell biology. So I do generally want you to know, the names of Hooke and von Leeuwenhoek and what they are associated with. So now moving forward to the 1800s, we have a couple more names who are synonymous with cell biology history. There is Matthias Schleiden, and he was a botanist. Right, so I was saying he was a botanist and pretty much was the person that decided that all plants were made of cells. Then there was Theodor Schwann, which is another famous biologist. He was the zoologist to Matthias's botanist, and he said that, well, guess what? All animals also made of cells. And then Rudolf Virchow said, you know what? All cells come from cells that already existed. So here I have another quick podcast style video called The Wacky History of Cell Theory, which is actually really interesting and a good way to get some of the background information on some of these guys. And So now we're going to talk about the cell theory, because at this point you already know this, that all living things are in fact made of cells. So cells are the basis of life. So the cell theory can be divided into three parts, if you will. And the theory states that all living things are made of cells. The cell is the basic structural and functional unit of life, and that all cells come from pre-existing cells. So when you think about it, these discoveries were made in the 1800s and they still hold true today. So now on your screen, you can see some examples of different kinds of cells. And actually, these are cells that come from every major kingdom that we classify life under in biology. So here we have a bacterium cell from kingdom Eubacteria. We have an archaean cell from kingdom Archaea bacteria. Here is a protist from the protist kingdom. Algae, which is also part of the protist kingdom. And then here we can see some fungal cells, which are parts of kingdom fungi. Then, of course, an animal cell. So this entire thing is the cell. This part would be the nucleus from kingdom Animalia. And here we can see some different kinds of plant cells from kingdom Plantae. Okay, so let's talk more on the structural level of cells. So cells are tiny. No life is smaller than a single intact cell. And a lot of things have to be achieved for them to be able to maintain life, including things like how do we get things in and out through diffusion? How much surface area do I have to allow for things to come in and out compared to how much volume does this cell contain, and how far will these things have to travel to get to the different parts of the cell? So cells cannot be viewed with the naked eye. Of course, we have to use microscopes in order to see them. And the ones that we do the most work with are the light microscope and the electron microscope. So I need you to now take your pen or pencil and get rid of this sentence. Your book will go over the light and electron microscope. I don't know the page off the top of my head. This went with the old version of the book, so it's been updated since then. Because cells are so small, we don't actually measure them in like millimeters or centimeters or anything like that. We have to measure them in microns or micrometers. And there is the unit for that. So because we have to magnify, we literally have to blow the cell up, make it many, many times bigger than it actually is in order to see it. When we're working with microscopes, the magnification of the microscope is important and the resolution of the microscope is important. But those things limit the microscope. Microscopes can only be so powerful, especially light microscopes. So even though I just showed you some very different pictures of types of cells, there's certain things that all cells are going to have in common, regardless of what kingdom it belongs to. So all cells are going to have a central area inside the cell where DNA is found. Excuse me. Remember, your DNA is that biomolecule that's made up of nucleotides that stores your hereditary information, your information for making proteins. Okay? That information is stored in segments known as genes. So those genes are found on the DNA, and they are wrapped up in what we call the central dogma of biology. And it means that DNA is used to make RNA and RNA is used to make proteins. When we get to protein synthesis, we will expand on that. All cells are also going to have a cytoplasm. The cytoplasm is everything inside of the cell, okay? And it's all the liquid and all the things floating in the liquid in the cell. It's going to divide the cell into areas that are structural, so holds the cell's shape, think of it that way, and areas that are functional, that do an actual job. Now, the cytoplasm itself is made up of solid pieces and liquid pieces. The liquid or aqueous pieces are known as the cytosol, so it's the liquid layer of the cell, just the liquid parts. And the solid parts are known as organelles. They are small structures, highly organized within the cytosol, and they all do very specific functions. So we will be delving into those specific functions in a little bit. Now, for some reason, it's cut off on my slide, but on yours, you should also be able to see that all cells will have a cell membrane, also known as a plasma membrane. And that cell membrane pretty much defines what is inside the cell from what is outside of the cell. So let's look at the cell membrane in a little bit more detail. And something that you will notice a lot in the images coming up is you will have the book cartoon drawing version of this image, if you will, and then there'll also be a picture of a real cell. And you'll realize that in real life, they don't look the way they do in the book, obviously. We make those pictures in the book highly colored and highly idealized, let's say, so that you can clearly tell things apart. But when you're looking at an actual cell, nothing's in color. It's all in black and white, and a lot of the structures don't look exactly the way they're drawn. So I like to show you as much as I can, hey, this is what the book says it looks like, and then this is what it actually looks like in real life. So that cell membrane, which is sometimes also called a plasma membrane, like I said, defines where your cell is. It defines or outlines the cell itself in terms of the cytoplasm. So it is a bilayer, a lipid bilayer, and this should be ringing some bells for you because we've already talked about this in our biomolecules unit. So that lipid bilayer is made up of phospholipids. So let me draw your attention to this portion of the screen, or really this whole portion of the screen. So here is my lipid layer, here is my top layer, and here is my bottom layer. And if I could pull just one of those out, this is what it's made up of. It's made up of a phospholipid, which is a type of biomolecule. Remember, the phospholipid is amipathic. It is dual natured. One part of the molecule likes to be in the presence of water, and the other part does not. So the head is the hydrophilic region. Another word for that is polar. It likes to be around water, whereas the tails are hydrophobic or nonpolar. They don't like water. They try to get as far away from water as possible. So the entire thing is a phospholipid, and each of these little orange balloons, if you will, represents a single phospholipid. So hundreds, if not thousands of them make up your cell membrane. Now, along with the lipids in the membrane, there are also proteins in the membrane. And you can see these proteins here, these blue tube-like looking things. So this would be a protein, and this would be a protein. And the proteins have various jobs, and we'll talk more about it when we get to the transport unit. Now, because such a big region, and when I say big, I'm talking this entire middle region that I'm coloring in green, all of this is actually hydrophobic. The majority of the bilayer is hydrophobic. So the plasma membrane itself is mostly hydrophobic. And because of that, it gives the plasma membrane the ability to be selectively permeable. Okay? So that hydrophobic region makes the membrane selectively permeable because only things that are similar in chemistry or similar in nature will actually be allowed into or through the membrane. So it helps to, like I said, distinguish your internal cell from the external environment that that cell is a part of, and it regulates movement. It regulates what enters and what exits. I always tell my high schoolers to think of it a little bit like the person at the front door when you go to a club or when you go to a concert, okay? Any and everyone can't just waltz in, even if you look the part. You have to have something to prove that you should be there, and they have the ability to turn you away. Plasma membrane plays a similar role. Okay, so now that we've talked structurally, let's talk about the types of cells that we can have. So we can take all life, all cells, and we can break them up into two big categories based on the presence of a certain organelle. Sorry about that. I thought my son was coming in. Hiding from my 4-year-old making this video in my closet. Dedication, I just want you all to know. Okay, so prokaryotes is one kind of cell, and eukaryotes are the other category of cells. So those are our two big categories, prokaryotes and eukaryotes. So if you are prokaryotic, you don't have a nucleus. So you still have DNA, but you don't have a membrane surrounding your DNA giving you a nucleus. So let's make a quick note of that. All right, so prokaryotes have no nucleus. We always teach the rhyme pro means no, meaning no nucleus. But they do have a nucleoid, a darker body within the DNA that's used for specific things, which we will talk about later on. There are two groups of life, two domains of life that fall under the prokaryotic cell umbrella. And they are both our bacteria. They are our eubacteria, sometimes we just call them bacteria, and our archaea bacteria. So on the flip side, so that's the big picture here is no nucleus. Okay. And then, of course, I would like you to know that archaea and bacteria are prokaryotes. Now, eukaryotes, and we think of the rhyme you do. So, eukaryotes do have a membrane around their DNA, so they have a nucleus. Okay, so these guys have a true nucleus. There is a boundary around their DNA, and all other life, if you're not a eubacteria or archaeobacteria, then you are a eukarya because your cells are eukaryotic. You have that nucleus. Now, there are a couple other things that we can look at to determine prokaryote from eukaryote, but that's the first hurdle that we cross. Is there a nucleus? If there is, they're eukaryotic. If there isn't, then they're prokaryotic. Okay, so let's look at a little bit more depth in terms of our prokaryotes. So another important thing to know is that our prokaryotes have no internal membrane structures. Okay, so in other words, they don't really have any organelles per se. They can have a cell wall and sometimes a capsule, another layer to give them added protection. And they do have a plasma membrane. And their plasma membrane actually carries out most of their metabolic functions. Now, with eukaryotes, you'll see this is a huge difference. While it still has a plasma membrane, we have organelles to carry out our eukaryotic cellular functions. And I say our because humans fall under the umbrella of having eukaryotic cells. Now, prokaryotes are going to have a nucleoid that has, and in that nucleoid chromatin, chromosomes. I missed something. I'm sorry. Now I know on my slide it's cut off, but you should have this on your slide if you printed these. So from here it goes on to talk about ribosomes. Now ribosomes do exist in prokaryotes. A lot of people consider them organelles. A lot of us in the biology community pretty much just think that they are a location at which something happens, but they are common to prokaryotes and to eukaryotes. So ribosomes are the only organelle that you find in both, and this is where protein synthesis, the making of proteins, takes place. Prokaryotes are going to have ways of moving their cells. Usually it's in the form of a structure called a flagella, so that's something important to note. And what exact structure just depends on the cell, because eukaryotes can have flagella as well. But I need you to know that flagella is used for movement. And then another thing to be aware of when it comes to prokaryotic cells, they're going to be fairly simple organisms, because think about it, bacteria are an entire organism, an entire living thing, and they're only one cell big. So they're pretty simple, but they're found everywhere. There is no place really that you can think of that bacteria don't exist. Some of them, the weirder, the more extreme, the better. Those would be our archaeobacteria. Sometimes we use the term extremophiles to describe them. So if we jump over to our picture really quick. That just did. Okay, if we jump over to our picture really quick, let's look at some of these parts and pieces. So here I'm going to have my plasma membrane and my capsid. All this purple swirly stuff in the middle, that's the DNA. Notice there's nothing surrounding that DNA, so that's why it doesn't have a true nucleus. All the little dots are going to represent my ribosomes, and then this is just my cytoplasm. I have this long whip-like tail. That is my flagella. That's going to help me with movement. And that's pretty much all that there is. Okay, eukaryotic cells. So these guys are the ones with the true nucleus. They are part of domain eukarya, and that's going to include everyone that isn't a bacteria. So all of my protists, my fungi, my plants, and my animals have eukaryotic cells. The eukaryotic plasma membrane is really just there to regulate and allow for the movement of substances in and out, and it also helps with cell to cell binding. Okay, so unlike prokaryotes, where that plasma membrane is where the majority of the metabolic processes take place, eukaryotes have evolved organelles to do their metabolic jobs. So the plasma membrane is just designed to help things come in and come out, and it also helps us to recognize parts and pieces in the living systems that they are a part of. Now, fungi, plants, and some of the protists are going to have cell walls, but everybody else doesn't. They just have the plasma membrane. Okay, so animal cells do not have cell walls. Important to know. All right, so here we're looking at a side-by-side with an animal cell. And this is our general representation of a eukaryotic cell. And you can see it looks so very different to the prokaryotic cell that we just looked at. First of all, there's a lot going on inside, okay? Lots of different parts and pieces. They all look different in terms of shape. They're hanging out in different parts of the cell. And it almost looks like there's a fair amount of compartmentalization, which is exactly what has happened with eukaryotic cells. We've designed these little sections for very specific reactions to take place there and only there. Okay, now if you look at it in comparison to a picture of a real cell, so here's my nucleus with my nucleolus in the middle. Everything on the inside here is going to be DNA. On the cartoon version, here's the nucleus, here's the nucleolus. And then you have a series of these little like circular style organelles, which are a variety of things. Okay, so these guys here are mitochondria. Here would be a mitochondria in the cartoon version. We have some endoplasmic reticulum here, which you can also see there. We have, see, here's a Golgi body. Here's one right here, and another one kind of here. That would be this structure here. All on the outside is my plasma membrane. Here's my plasma membrane. So you can see lots of parts and pieces. On the flip side, this is what a plant cell would look like. So still eukaryotic, still lots of parts and pieces, but some very big differences. So first of all, we have this structure that takes up a lot of space. This is called our central vacuole. And in plant cells, it helps to hold extra water so that those plant cells are nice and plump and they can actually stand upright. They can help the plant to hold itself upright. In a real picture of a cell, the vacuole just comes off as a giant space. Okay, so I always tell my high schoolers to think of your vacuole as a swimming pool. What do swimming pools do? They hold water. What does the vacuole do? It holds water. Okay, so that means that all the other structures kind of get pushed to the edge of the cell, and you can see that here. Now, one of the things that plants are known for is their ability to do photosynthesis. So these giant structures here, 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 that you can kind of see all around, those are going to be your chloroplasts. On the cartoon version, this is what they look like, and that's the location or the site of photosynthesis. So that's where they'll be actively making glucose. And then they still have all the other parts and pieces. There's a nucleus with a membrane. There's still going to be endoplasmic reticulum. We're still going to have rough ER. We're still going to have Golgi. All of the different organelles are still represented. They're just shifted around a little bit to accommodate that central vacuole. Okay, so now we're going to jump in and start talking about the individual organelles in eukaryotic cells. Excuse me. Some of them are standalone, and some of them work as parts of bigger systems. So we're going to start off with the main boss in charge. At least that's how we always describe it to little or younger students, and that would be our nucleus. So the nucleus was discovered by Robert Brown, and it was one of the discoveries that led to the cell theory. And the nucleus is defined by its nuclear envelope. So the nuclear envelope is the outer covering of the nucleus, and it separates the nucleus itself and its insides from the cytoplasm. Okay, so I'm going to number this as 1. On this picture, that would be right here. Okay, so if you zoomed in, that purple feature, I'm going to use my laser. So all of this, that is my nuclear envelope. Okay, it is a double membrane. It has 2 membranes. Again, look at the picture. Here is membrane 1. Here is membrane 2. Okay, so double membrane. A lot of these organelles are going to be double membrane. It's also not solid. So see these little green features that almost look like they have holes in the middle? Well, they are holes. They're pores, just like how you have pores in your skin. So those pores lead from the outside all the way into the nucleus itself. So those are some of the features that define your nuclear envelope. They're double membraned and they have openings that we call pores. So once you get inside, you have nucleoplasm. So that's everything that's floating around inside of the nucleus itself. And it's made up of some water, chromatin, and chromosomes. We'll talk about the difference between those later on. And then there is a darker feature, which I am going to number as 2. Oops. Oh, no. Sorry. Okay. I'm going to number it as 2. And here you can see it, 2. It's pointing right there. That is your nucleolus. And your nucleolus are the genes for creating a specific kind of RNA known as ribosomal RNA. Okay, so it's found inside the nucleus with all the other DNA. Okay, so from the nucleus, we'll next talk about the ribosomes. So ribosomes are the site of protein synthesis, the location at which protein synthesis happens. So a lot of people think of them as protein factories, and they're actually made up of 2 sections, a large subunit and a small subunit. And you can see those labeled on the diagram already. Now, ribosomes come in 2 forms. There are fixed ribosomes, meaning they're attached to something, and then there are free-floating ribosomes that are just floating around in the cell. So starting with the fixed ribosomes, they are fixed because they are attached to another organelle structure called the endoplasmic reticulum. And when the endoplasmic reticulum has ribosomes on its surface, it takes on this rough or bumpy appearance. So we literally call it the rough endoplasmic reticulum, or the RER. And we have to differentiate because there's another kind of ER that you'll learn about in a little bit. So if the ribosome is found on that endoplasmic reticulum, then it is going to make proteins that that cell is going to send away. Okay, so these proteins are going to be made and then they're going to be packaged off and shipped outside of the cell to be used elsewhere in the body, elsewhere in the system. Now your free ribosomes, they're just floating around in the cytosol, just hanging out, not bothering anybody, but they produce proteins that the cell will use for itself. So these proteins never leave the cell, they're utilized by that actual cell. So rough ER is going to make proteins for somebody else, but free-floating ribosomes, it's making proteins for the cell that it is a part of. Okay, really quick, if we look at the picture, so over here, you can see that these little red dots, I'm going to zoom in, or at least I'm going to try to zoom in. There we go. Oh, sorry. Not trying to make you seasick, even though I might have just accomplished that. Okay, so the little red dots are the ribosomes. See how they're attached to these tube-like things? That's your rough ER. And then if you look over here, you can see ribosomes that are not attached to anything. Those are your free-floating ribosomes. So fixed ribosomes, free-floating ribosomes. Over here in the plant cell, same thing. Attached to those tubes makes it fixed. Floating around randomly in the cell makes it free-floating. Okay, so the next several organelles, maybe not several, but the next few, let's say the next few organelles that we will talk about are part of a big system that we call the endomembrane system, so the internal membrane system of the cell. And what it does is it divides the cell into areas that are functional, so it does some kind of metabolic process, and then areas that are structural. It helps the cell to hold its shape so that that cell can live the life that it was designed to live. Now, these membranes can be connected either physically, so they're directly connected to each other, they're touching or they lead into each other, or they can be connected by making small little bubbles that we call vesicles that transport things back and forth from one part of the system to the other part of the system. So the membrane can be directly connected, so physically touching, or indirectly connected by vesicles, which are small sacs that transfer materials between different parts of the system. Okay, so the endomembrane system is made up of the following parts: the nuclear envelope, which is the outside of the nucleus, the endoplasmic reticulum, which actually comes in two forms. We've talked about one a little bit, but there's a second. The Golgi complex, sometimes called the Golgi body or the Golgi apparatus, same thing. Lysosomes, vesicles, and the plasma membrane. I guess I would have been right saying several the first time. So starting with the rough endoplasmic reticulum, or the RER, which we've already been introduced to. So again, it's rough because we have these ribosomes that can be found on the surface. The rough ER does take part in protein synthesis, but remember, these are proteins that will actually leave the cell and be used for jobs outside of that actual initial cell. It's used elsewhere in the body of that organism. And on the inside of the rough ER, that space is called the lumen, and it allows for those proteins to be transported around to the various parts of the cell so that they can eventually get packaged. And then the actual folds of the ER is known as the cisternae. Okay, and again, you can see cartoon version that shows everything in vivid detail. I can tell this is hollow. I can see the ribosomes on the surface, and these folds are the cisternae, as I said before, the space on the inside being the lumen. Now, if I look over here to my actual picture, then you have to look very closely. So all of these little, they're like little dots, if you will, that's the actual ribosomes. And then these spaces are your lumen. And of course, each fold is a cisternae. So the next part of the endomembranous system, and another one of the cell's organelles, is actually the smooth endoplasmic reticulum. So named because there are no ribosomes attached to its surface. Now, your smooth endoplasmic reticulum's job is to synthesize lipids that are going to become part of your cell membrane. So it makes those phospholipids. In the liver, in liver cells known as hepatocytes, smooth ER's job is to help with the detoxification that your liver is known for. So it converts drugs, poisons, and any other toxic products into substances that your body can either tolerate or that your body can flush out or remove from the body itself. So when you look at your smooth ER, the first thing that jumps out is that there are no ribosomes on the surface. Their tubes look a little bit more tubular, almost circular. The folds are still called cisternae, and the inside is still hollow, and that's still referred to as the lumen. All right, if you need to take a break, now would be a good time to do so. So feel free to hit pause and come back tomorrow, or come back in a couple minutes, hours, whatever it is you need to do for you. Okay, up next is the Golgi complex. So again, as I mentioned before, sometimes this is referred to as a Golgi body or a Golgi apparatus. Same thing, okay? Just know that they sometimes use those terms interchangeably. So the Golgi complex is another part of the endomembranous system. This is the post office, if you will, of the cell world. So your smooth ER has a very definitive function. Your rough ER is designed to make proteins and get those proteins folded up in the proper way so that eventually they can be sent out of the cell. It's the job of the Golgi apparatus to move these different proteins back and forth so that they're actually packaged and ready to leave the cell. So the Golgi apparatus is going to receive proteins through these membrane sacs that we call vesicles. And vesicles are just like, think of them as boxes that you would bring to your post office. So the vesicles are going to come from the ER carrying the proteins that the rough ER have already made. Once they get to the Golgi apparatus, we're going to put all the final touches. So they get modified, they get any additional parts and pieces attached to them, or any additional parts and pieces removed from them that shouldn't have been there. They get sorted depending on where they need to go. They get repackaged and reprocessed, and then they get sent to the plasma membrane and they are exported out of the cell. So very much like our mail service. Okay, so I always have my high schoolers picture, well, when I bring that card that I'm sending to my grandmother for her birthday, or that box that hopefully your mom will send to you when you go to college with care package stuff in it, all what has to happen, it has to get a label attached to it. I have to have the right address. I have to have any additional stamps placed on the box or the label. All of those things have to happen prior to the guy behind the counter taking it and putting it into the right bin so that it ends up in the right location. So it's the same situation here, but now we're just talking about proteins leaving a cell and not mail going to your grandma. So how exactly is export handled? So there are two ways that we can think about this. We can think about exporting things out of the cell, but we can also think about importing things into the cell. And the processes are called exocytosis, so literally out of cell, because exo is out, cyto is cell. And then there's endocytosis, inside cell. Okay, so in exocytosis, you are moving material out of the cell. So what's going to happen is that vesicle containing whatever it's containing, so in this case, let's say proteins, is going to travel up to the plasma membrane. And notice the vesicle is also made up of the same bilayer that makes up the plasma membrane. So the two are just going to fuse together, make the plasma membrane slightly bigger. It's going to open up so that whatever the material is can leave the cell, and then that material goes, if it's in like a human, for example, it'll get into the bloodstream and get transported to what other part of the cell it needs to go to. Now, just like how I can push things out of the cell, I can also pull things into the cell, and that process is the endocytosis. And it comes in two forms. There is penocytosis, known as cell drinking, used to move very small material. And then there's phagocytosis, known as cell eating, and that's used to bring in things that are very large. So it's the opposite process to exocytosis. We start off with the material right outside of the cell. That cell membrane is going to bend or fold in on itself, which makes this little pocket that you can see here. Once all the material has fit into the pocket, then the membrane is going to close off, so you can see how that bilayer is folded up to make what almost looks like a vesicle. And the vesicle gets pinched off. These pieces will rejoin to keep the plasma membrane whole, and then these pieces will come together to close off the vesicle, and it gets pulled into the cell and goes to whatever part of the cell it needs to go to. So this is a perfect place to talk about the lysosome, because a lysosome is also a vesicle, but it's a vesicle that contains very specific proteins that we call hydrolytic proteins. So these are proteins that are capable of breaking up molecules, and they exist in a very low pH. So when we bring material into the cell through endocytosis, whether that is phagocytosis for large material or penocytosis for smaller material, those vesicles get attached to a lysosome. And that lysosome helps to digest whatever that material is. So we especially see this in phagocytosis. Those enzymes are going to break up that material into its biomolecules and into its monomers, and then the cell can use those monomers to build new structures. Okay. This is also a major part of your immune system. So very often bacteria can get engulfed within a vesicle, and then these lysosomes will just destroy that bacteria so it's no longer hurting you, and we can use those parts and pieces of the bacteria for other things. Now, another role of the lysosome is in something called autophagy, where organelles that are past their prime, that are no longer functional or aren't working properly, can be destroyed and recycled, if you will. So really, your lysosome is kind of like the stomach of your cell. It's the thing that's going to do all of the eating, if you will, so that we have material we can reuse. Okay, so this picture kind of shows all of the endomembranous system, and you can kind of see how it's all connected. So we just kind of track through it a little bit. So here we have our nucleus. This region is going to make up our nuclear envelope that's directly connected to our rough ER. Remember, this is the rough ER because those ribosomes are on the surface of that rough ER. This tunnel system, if you will, is going to create proteins that will be exported via the plasma membrane to another location outside of the cell. Now, that rough ER is making these proteins, so you can see the little dots representing the amino acids. Here you can see they're all combining together. And then once they're all there, it's going to pinch off and make a vesicle, which will transport that protein to the Golgi apparatus. Remember, the Golgi apparatus is where all of the final modifications take place, and then they get sorted into whatever part of the cell it needs to exit from, becomes a vesicle again, and that vesicle transports all the way to the plasma membrane and then pushes that material out of the plasma membrane. Now, if material coming in, like for example, here we have this old organelle that's damaged, or we have material that came in from the plasma membrane, our lysosomes are going to associate or fuse themselves to that vesicle carrying the organelle or carrying the material, and it's going to recycle all of those monomers so that the cell has more options for making more biomolecules. Okay, up next is the mighty mitochondria, what everyone tends to remember from high school. So your mitochondria's job is to perform the metabolic process known as cellular respiration. And cellular respiration is going to produce ATP, cellular energy. The mitochondria structurally has two membranes, an outer membrane which is smooth, and then an inner membrane which is folded. Inside of those folds is the liquid in the mitochondria that we call the mitochondrial matrix. Okay, so I've written a few additional notes there for you, and then I've also labeled where you can find these various parts and pieces on the diagram. So the mitochondria is an interesting organelle because while it fits the bill of an organelle, it also suggests that at some point this organelle wasn't just an organelle. So what do I mean by that? Well, the mitochondria has its own DNA. It has its own genome. And it also has its own ribosomes. So why would an organelle living inside of a cell need its own separate DNA that has nothing to do with the DNA in the nucleus, and its own way of making proteins? Well, that leads us to a theory that has been ongoing for many, many, many years of endosymbiosis, which is one of our theories that kind of grounds evolution for us. So we'll talk more about that later on. So what is cellular respiration? So cellular respiration is the process of making ATP, and it's done using oxygen and glucose. And the oxygen and glucose get converted into water and carbon dioxide. And while that conversion is happening, cellular energy in the form of ATP is also done or formed. Okay, another set of organelles that people don't very often talk about, they're not very mainstream, are called microbodies. So, microbodies are single membrane organelles, they only have one layer. They are not part of the membrane system, and they have enzymes that help to link some of the different metabolic pathways or biochemical pathways. So examples of microbodies, are things like peroxisomes, glycosomes, or glycosomes, and a lot of them help with the detoxification of the cell. Next is the cytoskeleton. So the cytoskeleton does exactly what the name suggests. It is an internal skeleton that helps to maintain the shape and organization of the cell. And it's made up of these interconnected protein fibers and protein tubes. You see it most often, it's most prominent in animal cells, but plant cells and fungi tend to get their support from their cell wall and their central vacuole, so they don't need them as much. Does it mean that they don't have them at all? No, they're just not as rampant, let's say, because they have other ways of keeping their cell shape. And their cell shape is actually very specific, whereas with animal cell shapes, they change depending on the environment. So here you can see three examples of the cytoskeleton: the microtubule, the intermediate filaments, and the microfilaments. Okay, so let's look at these cytoskeleton pieces in more detail. So the main component of animal cytoskeletons are microtubules, intermediate fibers, and microfilaments. Your microtubules are supportive in nature. They're made up of a material called tubulin. Your intermediate fibers I veered off. So microtubules, because of their role as being supportive, they tend to be bigger and thicker. They look stronger, okay? Intermediate fibers are thinner and are interconnected. They're interwoven between these microtubules. And then, so they sometimes referred to as just intermediate. And then your microfilaments, let the words guide you, micro meaning tiny, small, thin, whatever, are the thinnest of the three, the most prominent being one called actin. So if you look at the pictures, it makes sense. So here are my microtubules, big, takes up lots of space. Yes, they're hollow on the inside, but notice that twisted spiral shape. Then we have our intermediate filaments, okay? So each of these lines is an intermediate filament, and then when you look at them even more closely, they have this double helix appearance, but they're not DNA. And then lastly, we have the microfilament, who are smaller, made up of these actin pieces, and they're kind of twisted together like it's a very gentle spiral, a very gentle turn. So the microtubules that we really care about are the ones that will help your cell to divide. So a lot of them are going to initiate from a structure known as the centrosome, and that originates from centrioles. It's their job to anchor or hold major organelles into place, which would make sense because some of the organelles are pretty big, so it makes sense that the biggest of these cytoskeleton pieces is what's going to hold something in its location. And then microtubules also provide roadways or pathways, tracks, if you will, for the organelles that actually move, because some of them are mobile. Excuse me. Now, the organelles that move don't move by themselves. They move with the help of a protein known as a motor protein, and that process is going to require ATP or cellular energy. So, your cytoskeleton allows a large amount of cellular movement, especially for your cells or your organisms that are just a single cell. Okay, they can't just sit in one location. They actually have to be able to travel. So if they don't have cilia and flagella, then they actually have to move their entire cytoplasm in something known as amoeboid motion or cytoplasmic streaming. And like I mentioned before, when we divide, when cell division happens and we have to move chromosomes around, it's your microtubules that are also doing the bulk of that work. So like I mentioned before, some cells have extra features like cilia and flagella that allow them to move, that aid in movement. Their structures are the same, but cilia are shorter and more plentiful, and flagella are longer and fewer. Okay, the cilia and flagella are formed from microtubules, and they have what we call a 9+2 arrangement, which you will see in a little bit. So it's 9 sets of 2 molecules repeated over and over again, almost like in a circle. Okay, and a lot of them are found in motor proteins, the things that are going to allow those organelles that do move to actually move. And prokaryotes have cilia and flagella, but they are not the exact same. Okay, one important difference between your cilia and flagella is how the movement takes place. So flagella are long tail-like structures. Think of like the tail of a sperm cell, for example, and their movement is more ondular. So up and down and up and down and up and down. So you get this waving motion that pushes the cell forward. Whereas your cilia move in the same direction. Think of it as we're rowing a boat. There's 7 of us in this rowboat, and we're all rowing at the same speed, in the same sequence, in the same direction. So each little hair is going to move the same way. We're going to push out and pull back in. Push out and pull back in. Push out and pull back in. Okay, so now we're going to get to an organelle that is specific to plant cells. You're not going to see these in animal cells. And these are chloroplasts. So your chloroplasts are also double membraned. Actually, you can think of them as having multiple membranes. And that really helps because their job is to make food in the form of sugar through the process of photosynthesis. So they just like the mitochondria, they have an outer smooth layer, and then they have inner folded layers. They also have liquid on the inside, and that liquid is known as stroma. And their inner folds are known as the thylakoids, and then collectively as the grana. So let's see if I can make this obvious. I guess we'll go with red. So one single fold, and these are actually like discs, like little flattened discs. To be honest, I always think of pancakes when I think of chloroplasts, because if you think of a short stack of 5 pancakes or 3 pancakes, however much is in a short stack, one on top of the next, that's what these little discs look like. They come in piles of several on top of each other. So this that I just outlined, that would be one thylakoid. This would be another thylakoid. Okay? But when we talk about the entire stack, so let's call that 1, put a 1 there. When we talk about the entire stack, like we are doing here, this entire thing, that whole outline, that is a grana. So let's call that 2. Okay, so the thylakoid is the individual disc or pancake, if you will, and then the grana is the entire stack of discs or the entire stack of pancakes, if you will. Now, just like with your mitochondria, these guys suggest that they once lived on their own. And it's because they have their own separate DNA and their own separate ribosomes, just like the mitochondria did. So that is another point of support for that theory of endosymbiosis. So plastids are organelles that are specific to plant cells that can include chloroplasts, but also include amyloplasts and chromoplasts, and they all do a similar function. They help with food production. Okay, so up next we have the central vacuole, again specific to plant cells, and we've already kind of introduced these guys. So these are just really large vesicles that make up the majority of a cell's volume. About 90% of the cell's volume is going to be found in that vesicle. They help to put pressure, what we call turgor pressure, onto that cell, especially the cell wall, from the water that plants absorb. So by that central vacuole being completely full, that pushes on all parts of the cell, making the cell stand up straight, and that cell pushes on the next cell so that your plant stands up straight. So this is why when you don't water your plant, they wilt, because the first thing to go is going to be that water in the central vacuum, and then the cell almost collapses in a little bit on itself. So that's its main function, is to help with structure. And then of course it also stores sugars, salts, proteins. It can sometimes store waste material. It can contain chemical material that the cell would use against things that can make it sick. And then it also helps with the enzymatic digestion of food material. Okay. Now, the membrane that surrounds the vacuole has its own name, because this is biology and we love to name things, and that is known as the tonoplast. So the tonoplast is the membrane that surrounds the central vacuole. The vacuole itself is that large vesicle found on the inside of cells, of plant cells, sorry. Okay, up next we have the cell wall. So plant cells and a few of like some fungi and stuff like that, bacteria as well, also have cell walls, but animal cells do not. So plant cells have a cell wall and a plasma membrane. Your cell wall is the most outward structure. It's an extracellular structure, and it helps to provide support and also to contain that pressure from that central vacuum being full of water. In plants, it's made up of cellulose fibers because they are very strong, and then it also can have other organic materials in there to help with its job of keeping structure. So there are two kinds of cell walls. There are primary cell walls and secondary cell walls. The secondary cell wall is actually found closest to the plasma membrane, and then the primary cell wall is found on the outside of the secondary cell wall. There is another feature or structure. Hold on one second. Okay, I just wanted to put that note in there for you. Now, because plant cells are found stacked on top of each other, there's actually another layer that holds two adjacent cell walls together, and that is known as the middle lamella. Now, there are times where plant cells have to be able to communicate cell to cell or to pass material back and forth. So they are actually interconnected cellularly by spaces or junctions that we call plasmodesmata. Okay, and those plasmodesmata have no cell wall passage at all. So if you look at the picture right here, see these little gaps? That's your plasmodesmata. There's no cell wall inside of the gap, so that allows the cells to send signals to each other back and forth. So in animal cells, they have these features called cell junctions, and it's the job of, well, let's put it this way. There are times where your cell has to be able to connect with another cell. And there are times where this needs to be a permanent situation and times where it needs to be a non-permanent situation. So when it's non-permanent, it's referred to as cell adhesion. And the cells just bind together temporarily. And then after they have exchanged their information, they can unattach themselves. And the way they hold on to themselves is they have glycoproteins on their cell membrane that are specific to molecules on the other cell. So the two fit together and they hold the cells together temporarily. Now, the opposite of that, the permanent situation, is called a cell junction. And a cell junction is a space in between cells that's sealed, but it's a permanent connection. And it's used in something called direct cellular communication. Cell A needs to speak to cell B and only cell B, so they are interconnected with each other permanently. So in terms of how this happens, well, there are three kinds of permanent attachments. There are anchoring junctions, and those literally like stick the cells together. So think of like welding, where you heat up the piece of metal and boom, you use that metal to connect two things. So same thing here. There are materials that hold those two cells together. They're called desmosomes and adherents. Then we have tight junctions, which prevent small ions from moving in between, but other things can pass. Okay, so again, sealed space. And the membranes are actually fused. So think of it as one giant cell sharing a membrane. So this is what I mean by that. So here I have, actually, I'm pretty sure the picture coming up does a way better job than anything I could draw. My high schoolers would tell you I'm not a good artist, and they're not wrong. Okay, and then lastly, we have gap junctions, which allow things to move through without the membrane having any control. And this is normally found in cells that are all the same, doing the same job, so they are a tissue. Okay, so here you can see all three represented. So here we have our anchoring junctions, and we just have regions where the cells that are next to each other can communicate with each other, and they're held together by these materials on either side of the cell. Okay, then we have tight junctions up here at the top, and again, connections between adjacent cells. Excuse me. Excuse me. And this allows for their plasma membrane to be connected. Okay. And then lastly, we have gap junctions, which are pictured here. And of course, here are the blown up versions of the same thing. So these allow for direct channels from that cell to the cell next to it. And it allows small materials to pass back and forth. Okay. Animal cells can sometimes secrete a material that's known as the extracellular matrix. That extracellular matrix is a combination of a couple of things, but most importantly, glycoproteins are the main component. And then it's not water, but there's a gelatinous, think jello-like material that's actually polysaccharide based. So it's a sugar, and we call that a ground substance. And then we have protein fibers. And all three of those things make up the extracellular matrix. So what's the job of the extracellular matrix? Well, it helps to provide support for the cell, and it also helps these cells to stick together. Cells that are the exact same, that are doing the same function, are known as tissues. And tissues have to be able to be very close to each other so that they can be maximally efficient. So that extracellular matrix allows them to stick to each other. Okay, it's called the extracellular matrix because it's found outside of the plasma membrane. So it's like glue on top of the cell, if you will, or in between individual cells. So in terms of the protein fibers, there are really three of them that play a central role. So there's collagen, which everybody's all the rave about collagen right now, and drinking collagen and putting it in your smoothie and taking the capsules, and more power to you. But collagen is known for being super elastic, which is why our skin stretches and goes back to its original shape, at least until we get to a certain age that happens. But it's also a very strong material. And then we have proteoglycans, which help the cells to interlink with each other. And they can go anywhere from being very jelly-like, so almost like jello, to being hard and elastic depending on what that tissue needs. And then we have fibronectins, which help to connect your cells via something called an integrin. So here is an image of what the extracellular matrix looks like. So here is my cell. So all of this is on the outside of the cell. And you can see they're just various types of proteins in different appearances, all floating in this jelly-like material, but helping to stick cells together so that they can be very functional and efficient. Okay, and then lastly, we have what should have been a video of a fluid mosaic, just kind of showing how the plasma membrane is made up and how it behaves. And I will have this linked for you in the lesson. All right, y'all. I will talk to you soon. Bye.